Today we are going to learn about structure and germination of seeds. Let's start. Let us study about seed. Seed is a mature ovule after fertilization. It contains a tiny living plant called the embryo. The embryo remains in an inactive state until it is exposed to favorable conditions, then it germinates. The seed also contains food material for the nourishment of the embryo during germination. The embryo can withstand unfavorable conditions of temperature, drought, etc. Do you know what? I hear some seeds are dormant up to more than 100 years. Classification and structure of seeds. Types of seeds by cotyledons. There are two types of seeds by cotyledons. They are monocotyledon seeds, seeds with only one cotyledon, and dicotyledon seeds, seeds with two cotyledons. And examples of monocotyledon seed is maize and grass, and dicotyledon seeds examples are gram and bean. Next, let us see the types of seed by size. Some are very small that they are barely visible to naked eye, like example poppy seeds. Some are quite large, like as in a watermelon, pumpkin and mango seed. Some are the largest are those of coconut and double coconut. Types of seeds by endosperm. Examples: albuminous, cotyledons are membranous, and endosperm persists. That's why we call it as albuminous, right? Example: monocot albuminous seeds and poppy and custard, dicot albuminous seeds and cereals and millets. X-albuminous, cotyledons store food and thick. Endosperm absent, that's why we call it as X-albuminous. So examples: monocot X-albuminous seeds, Amorphophallus and orchids. Dicot X-albuminous seeds, gram and pea. Now let's study about the bean seed. The seed cone is divided into testa and tegmen. First, let us study about testa. Testa is the outermost hard brownish covering. It protects the delicate inner parts of the seed from injury, lack of bacteria, fungi and insects. Next, let's see about the tegmen. Tegmen is also a thin layer and also protective like testa. Next, let us study about the hilum. Hilum is a whitish oval scar on the concave side of the seed. It represents the spot where ovule, which is now a seed, was attached to the ovary through placenta. Next, let's study about the micropyle. Micropyle is the opening through which the pollen tube had entered the ovule. When soaked in water, seeds absorb water mainly through this micropyle and make available to the embryo to germinate. It provides for the diffusion of respiratory gases for the growing embryo. On carefully separating the two cotyledons, you can see a tiny embryo. Tiny embryo consists of a radicle which forms root and the plumule which forms shoot. Next, see about the maize grain. Fused pericarp and testa. The pericarp and testa are fused in a maize grain and therefore it is called as a grain, right? The remaining major part other than embryo presents the endosperm. The endosperm is rich in starch, occupies major part of the grain. The separation of endosperm and the embryonic part was done by the ellurin layer. See, here you can see detailedly where the ellurin layer presents. The ellurin layer is rich in proteins, separates endosperm and embryonic part. The embryo is a tiny growing new plant which has two parts, radicle and plumule, and also consists of a cotyledon, that is scutellum. Embryo consists of a single cotyledon named scutellum, right? Remember scutellum. Plumule is a rising shoot and has a protective sheath called coleoptile. The radicle, a rising root, has a protective sheath called coleorhiza. Remember, radicle has a protective sheath called coleorhiza and the plumule has a protective sheath called coleoptile. Now let's know the differences between a bean and a maize. Germination. The seed contains a dormant embryo. It is said to be in a state of dormancy. The chemical activities which are going in it are slow and little food is used. When placed under proper conditions, the dormant embryo awakens and it becomes active and grows into a seedling. All the changes leading to the formation of seedling collectively called germination. Definition of germination. Germination is a process of formation of seedling developed from the embryo. Next. Do you know a fresh seed from the plant normally does not germinate even if the conditions for the germination are favorable? It must pass through a period of dormancy during which it undergoes physiological maturation. Now, let's see the conditions which are necessary for germination. The conditions necessary for germination are water, temperature, and oxygen. First, let us study about the water. The seed obtains water from soil. The water is absorbed all over the surface but mainly through microbial. Uses. The seed swells and consequently the seed coat ruptures, allowing the elongating radicle to come out and form root system. Water is necessary for chemical reactions and for the enzymes to act upon the food stored in the cotyledons or endosperm so that it may convert into a diffusible form dissolved and utilized by the growing embryo. The second one is the suitable temperature. Both high and low temperatures are not good for germination. Very low temperature inhibits the growth of embryo. Very high temperature destroys delicate tissues. A moderately warm temperature like 25 degrees to 35 degrees Celsius is usually favorable. So that's why it is called the optimum temperature. Seeds of tropical plants often need a higher temperature for germination than those of temperate regions. Remember, this point is very important, the last one. The next one is the oxygen. During germination, there is a rapid cell division and cell growth for which energy is required. The energy is available only by respiration and hence the need of oxygen. Do you know what? When you sow the seeds deep inside the soil, it fails to germinate. Do you know why? Because there is no supply of oxygen and insufficient pushing force in the embryonic part, that is epiglottal and hypocotyl, to break through the upper layers of soil. Now let's see some experiments on germination. Experiment to prove water is necessary for germination. Take two bakers A and B. In baker A, place some seeds of green gram or etc. on dry cotton wool. Similarly, in baker B, place some seeds with wet cotton wool. Keep both bakers in an ordinary room. In 1 or 2 days, you can see the seeds of baker B germinated while the seeds of baker A not. Because in baker B, we put a wet cotton wool. This proves that the water is necessary for germination. See this diagram below. Experiment to prove suitable temperature is necessary for germination. Again, take two bakers A and B, place some green gram seeds on wet cotton wool in each of the two bakers. Keep baker A in an ordinary room and baker B in refrigerator. In 1 or 2 days, the seeds of baker A starts to germinate, showing the importance of suitable temperature for germination. The seeds in baker B does not show any signs of germination or may germinate after several days, though not to extend the seeds in a baker A germinate. Experiment to prove oxygen is necessary for germination. Take two conical flasks A and B, spread wet cotton wool in each flask and place on it. When soaked gram seeds, lower a small test tube containing alkaline pyrogallic acid which absorbs oxygen. In flask B, by means of a thread, taking care that single drop of chemical falls on the seeds or the cotton wool. Keep the tube hanging by fixing a cork on the mouth of the flask. Arrange flask A in the same way except the test tube in the flask contains plain water. Place two flasks in an ordinary room. The seeds of flask A will germinate, showing the importance of oxygen for germination. The seeds in flask B does not germinate because there is no oxygen. There may be very slight germination due to anaerobic respiration in the absence of oxygen. Next, the 3 bean seeds experiment. In this experiment, 3 mature air-dried bean seeds are taken and tied to a glass slide at 3 positions as shown in the figure. This slide is kept in a beaker containing water in a manner that the top seed is well above the water level and the middle one is just at the water level, means the surface, and the bottom one is deep in the water. The experimental setup is left in a warm place for a few days, and the result here goes. The middle seed germinates as it gets both oxygen and water and also temperature. The top seed does not germinate at all as it receives only oxygen but no water. The bottom seed does not germinate or stop germinating after the emergence of a small radicle. It gets water or very little oxygen from the air dissolved in water. This experiment proves that the water is initial for germination, but the other requirement, oxygen, is not fully demonstrated. Types of germination. Knowing the types of germination, let us know the differences between what is hypocotyl and epicotyl. First, let us know what is the cotyl region. The region with the cotyledons attached is called the cotyl region. Epicotyl. The region of the axis between the point of attachment of cotyledons and the plumule is called epicotyl. Now, let's learn about the hypocotyl. The region of the axis below the cotyledons is called hypocotyl. Nothing is there. Remember, you know the cotyl, right? Where the cotyledons attach is called cotyl, and above the cotyl is called epicotyl. Epi means above, and the below is called a hypocotyl, which means below. Remember, both hypocotyl and epicotyl never elongate together. Either hypocotyl is elongated or the epicotyl is elongated. Now let's see what happens if one of them is elongated. Hypogeal germination. If the epicotyl elongates, the cotyledons remain underground. On the ground, if seed is just on the ground, and the germination occurs only in monocotyledonous seeds. Remember, this is very important. And then the type of germination is called hypogeal germination, P and gram. Let's see about epigeal germination. If the hypocotyl elongates, the cotyledons are pushed above the ground and the germination occurs only in dicotyledonous seeds, then that type of germination is called epigeal germination. Example, castor and bean. Difference between epigeal and hypogeal germination. Germination in some common seeds. Pea seed. The seed absorbs water and swells considerably. The testa softens and bursts, allowing a small radicle to form. The radicle emerges, grows downwards and forms the root system. The plumule grows upwards and forms the shoot of the seedling. In earlier stages of development, the plumule is arched and thus protects the young shoot from injury during its emergence from the soil. The cotyledons supply food till the seedling is able to exist independently. The cotyledons remain underground and the germination is thus called hypogeal. Next, let's see about the bean seed. The seed absorbs water and swells. The radicle grows and forms root system. The radicle grows out large, bursting the testa. The arched hypocotyl grows, forming an arc above the soil. It then straightens, bringing the cotyledons above the soil. The cotyledons become the first green leaves and soon fall off after the foliage leaves grow. The germination that is called epgl germination. Next, let us see about the maize grain. The seed imbibes water and swells. The radicle pieces to the protective root shield, coryza, and the fruit wall starts to grow downwards to form the root system. The plumule pieces to the protective root shield, coleoptile, and grows straight upwards. The growing fruit wall dies off soon, and small root grows from the fruit wall. The cotyledon scutellum absorbs food from the endosperm till it is exhausted. The hypocotyl does not elongate. This germination is called the hypocotyl germination, where the cotyledons are remain underground. Next, let's see about the viviparous germination. The special type of germination which shows in some mangrove plants like Rhizophora and Sonacea, in which the seed germinates inside the fruit where it is still attached to the parent plant. After germination, the plant drops the seedling into the soil, which develops root and fixes itself. Hi, I have given some questions and in the description below and in the comment box. Please fill the MS Forms link and practice. Thank you.